Okay, so good afternoon. So for today, we will be continuing our lesson on the anaerobic bacteria. So as a medical technologist, it is very important for us to know the proper procedures on processing colonies suspected of being anaerobes because during the part one, we have actually mentioned when to suspect that the colonies are anaerobic bacteria. Now, to confirm your suspicion, these are the steps that you need to undertake. So first, you need to describe the colony morphology and growth on selective media. So that is very important so that you'll be able to eliminate unwanted microorganisms by the virtue of a particular inhibitory substances being added on the selective medium. And then, of course, in order for us to categorize them, we need to describe the Gram-Stain reaction and cell morphology. And in order to further affirm our suspicion, then we have to do the aero tolerance test. Air tolerance test is actually a procedure to determine whether the organisms are able to tolerate oxygen. So if they are really anaerobic bacteria, microorganisms will not be able to tolerate oxygen. And of course, we need to inoculate the pure culture or eventually subculture them and add the appropriate disk in order for us to confirm whether we are really indeed dealing with anaerobic bacteria. So this is an example of anaerobic blood agar plate from intrauterine device via dissection microscope. So when you see dissection microscope, we are actually pertaining to scanning microscope. So in using scanning microscope, we can actually place the entire plate and then we can actually um, see the colony, okay, the colony under the microscope. So between so that we'll be able to see the magnified appearance of the colony. So this is a very good device. Okay, now let us discuss the schematic diagram for initial identification of anaerobic isolates based on gram stain morphology. So here, we can divide anaerobic bacteria into two major categories, whether they are gram positive or whether they are gram negative. Okay. So, if they are gram positive, of course, we classify them whether they are bacilli or cocci. And if they are bacilli, we further categorize them whether they have spores or they are non-spore formers. So, incidentally, one of the most medically important anaerobic bacteria are those that are gram-positive spore forming. Okay, so this would Clostridium species, the different species of Clostridium will fall under this category. Now we also have anaerobic non-spore forming bacilli. So various genera of bacteria will fall under this. So this would include Actinomyces, Bifidobacterium, Cutibacterium, Egertella, Eubacterium, Lactobacillus, and Propionibacterium. Okay, so these are the gram-positive anaerobic bacilli. Okay. And then we also have the gram-positive anaerobic cocci. Okay, so this would include Anaerococcus, um, Finegoldia, Parvimonas, Peptococcus, Peptoniphelus, Peptostreptococcus. So these are all classified as gram-positive anaerobic bacteria. And then we also have gram-negative anaerobic cocci. So we have Veillonella under this category. And we also have gram-negative anaerobic bacilli wherein we can find Bacteroides, Fusobacterium, Parabacteroides, Porphyromonas, and Prevotella. Okay, so these are the major categories based on gram stain. So that's why it's very important to categorize them using a gram stain. And then, as what I told you before, aero tolerance testing is very important because this will determine whether a microorganism isolated under anaerobic condition is either strict or facultative anaerobe. So when you say strict anaerobe, it means that they are obligate. meaning to say they cannot survive, okay, the presence of oxygen. But here, when you say facultative anaerobe, so they are aerotolerant, meaning to say they can survive oxygen, but they are anaerobic by nature. So we also inoculate them with a short streak onto a chocolate agar plate for incubation in a CO2 incubator and anaerobic blood agar incubated anaerobically. Okay, so that's one way of doing it. Okay, so here we can have various categories of aero tolerance test results. Okay, so based on the aero tolerance test results, we can actually classify them into four. Okay, whether they are obligate aerobe, capnophilic aerobe, facultative anaerobe, and obligate anaerobe. So when you say capnophilic aerobe, it means that this particular group of bacteria would be carbon dioxide loving. So if you have actually done this procedure, blood agar plate incubated aerobically in a non-CO2 incubator, you wouldn't expect obligate anaerobe and capnophilic aerobe to grow. Because why? Because you incubated it aerobically, so they will not grow. And then it is a non-CO2 incubator, so capnophilic aerobe will not grow. However, if you will be using chocolate agar incubated aerobically in a CO2 incubator, you would expect growth in all of the four categories except again for obligate anaerobe. Because remember, you actually incubated it aerobically. But if you will be using blood agar plate incubated anaerobically, you would expect growth in for facultative anaerobe and obligate anaerobe. But no growth here because you didn't include it, you didn't place it in a CO2 incubator, and of course, no growth for obligate aerobe. But it says here, if you will be reading here, it says here Haemophilus influenzae will grow on an anaerobic incubated Brucella blood agar plate, but can be differentiated from anaerobe by its growth on chocolate agar in a CO2 incubator. So for Haemophilus influenzae, which is this one is a special it has a special characteristic. So it will grow here, and at the same time it will actually um, grow here. But you have to use, instead of chocolate agar, you have to use a Brucella blood agar plate. Okay, so this is how we do the aero tolerance test. Okay, so I hope that's clear. Now, let us discuss the presumptive identification of some clinically significant anaerobic bacteria. Okay, so one of the tests that we can possibly do is fluorescence test. Okay, so for the fluorescence test, we are using the ultraviolet light. Okay, so we are using the ultraviolet light for Pheromonas and Prevotella because for Pheromonas and Prevotella, there would be a brick red fluorescence. Okay, so later on, I will be showing you the picture of how it looks like. And then, Fusobacterium nucleatum, Clostridium difficile, chartreuse fluorescence. Okay, so on the next slide, so we'll see how does a chartreuse look like. And Villanella would have a red fluorescence, and it is highly dependent on the culture medium. So the requirement here is utilization of an ultraviolet or the UV microscope. Okay, so here is an example of brick red fluorescence. That is for Pheromonas and Provotella. And Fusobacterium nucleatum and Clostridium difficile will have the chartreuse fluorescence. So this is the chartreuse. This is the chartreuse fluorescence for that particular bacteria. Okay, so these are the different types of fluorescence under long wave ultraviolet light. Okay, so for those Prevotella that are pigmented, okay, it will produce brick red. Prevotella, vivia, and desciens, it will produce light orange to pink. For Porphyromonas, for Porphyromonas, it will result to brick red, but no fluorescence for some. Fusobacterium, chartreuse. So chartreuse uh, is actually similar to yellow green, but with a harder spelling. Veilonella is red, but it fades rapidly. Clostridium ramosum is somewhat red. Clostridium inoculum and difficile will be chartreuse. And Agartella lenta would be red or sometimes no fluorescence. Okay, so this is just a guide for the presumptive identification. So we say presumptive because it is not confirmatory. So other tests would include catalase test, 15% hydrogen peroxide added to organism on slide. Bubbling is considered to be as positive. So it separates Clostridium from Bacillus because Bacillus is expected to be positive while Clostridium is expected to be negative. Now, this one is an example of the spot indole test. So this is a positive result and then this is a negative result. So incidentally, in the spot indole test, we are actually using DCMA reagent, which stands for dilated cardiomyopathy with ataxia. Okay, so we placed filter paper saturated with paradimethyl aminoxinamaldehyde, and if it turns blue green, okay, that is considered as a positive, whereas pink or orange is considered to be as negative. And then the rapid urease test and rapid motility test. Okay, so these are the other tests for presumptive identification of anaerobic bacteria. Okay, now this test, we are using this test as a special potency antimicrobial disk. Okay, so all we have to do is to add this particular disk. Okay, if this is the culture, we have to add a particular disk and see whether there would be zone of inhibition. Okay, so in zone of inhibition, so the presence of the zone of inhibition is actually tantamount to susceptibility. And susceptibility means that it is a positive reaction. So again, absence of zone of inhibition. So this is interpreted as resistant. And it is interpreted also as a negative reaction. Okay. So for vancomycin disc, susceptibility indicates gram-positive bacillus or Porphyromonas. For kanamycin disc, susceptibility indicates the presence of Bacteroides and other Fragilis group, also Bilophila, Fusobacterium, and Veilonella. For colistin disc, susceptibility indicates the same result or the same interpretation as kanamycin, but we include also Prevotella for this particular sensitivity test. Okay. Now that we are familiar with the sensitivity disk, so we can actually add this particular disk depending on the gram reaction and morphology of the bacteria that we have isolated. So for example, if we are dealing with gram-positive cocci, so it is expected that we should be using the SPS disk and nitrate disk. If we are dealing with gram-positive bacilli that appears like Clostridium, how would you say it appears like Clostridium? Perhaps because of the presence of spores. Then we don't use any of these at all. But if uh, we are suspecting or dealing with gram-positive bacilli that are branching or appears like or diphtheroid-like, then we have to use nitrate disk. If we are dealing with gram-negative cocci, then we can use the nitrate disk. And if we are dealing with gram-negative bacilli, Okay? Then we have to use, madami to, vancomycin disc, kanamycin disc, colistin disc, nitrate disc, and even the bile disc. Okay, so this would be useful for presumptive identification of anaerobic bacteria. Okay, so here's an example of kanamycin disc um, that is positive because of the presence of the zone of inhibition. This is the vancomycin disc that is positive because of the presence of zone of inhibition. And then this is the colistin disc that is considered to be as resistant because there is no zone of inhibition. Okay, so this is an example of the special potency antimicrobial disc results for Clostridium ramosum. Okay, so how do we interpret? So if the organisms are susceptible to vancomycin, okay, has a varied result in kanamycin and resistant to colistin, probably if it's pink staining, we are referring to Clostridium ramosum or Clostridioforme. However, if the kanamycin is resistant, it could be Porphyromonas. Kung if ever lang na resistant to, it could be Porphyromonas. Katulad nito. Okay? So the only difference between result 1 and result 2 is actually kanamycin. Okay, result number 3 means that all of the Ds are resistant. So probably you are dealing with the member of Bacteroides fragilis. But it could also be Prevotella or Parabacteroides. So again, it's not confirmatory. It is just for presumptive identification. Test result number 4 means that you are resistant with both vancomycin and kanamycin, but you have a variable result with that of the Prevotella. Okay? With that of colistin. So it means that you are dealing with Prevotella. Result number 5 means that you are resistant in both vancomycin and kanamycin, but you are susceptible in colistin. So the organisms are susceptible in colistin. So it means that most probably you are dealing with Prevotella. But if you are, but if the organisms are susceptible in both kanamycin and colistin, but resistant to vancomycin, it means that uh, you are dealing with the following bacteria: Campylobacter uroliticus, Bilophila wordsworthia, Fusobacterium, and even Veillonella. Okay, so these are the different antimicrobial or different ways to interpret special potency antimicrobial disc. Okay, so a while ago I did mention about nitrate, SPS, and bile disc. Okay, so nitrate disc, um, it's actually the same as nitrate reductase enzyme or nitrate reductase test because nitrate test will determine the ability of the organisms to reduce nitrate to nitrite. SPS stands for sodium polyanethol sulfonate. Okay? Actually, SPS is an anticoagulant that we're using for blood culture, but in this case, it is not an anticoagulant, but it is incorporated in the disc. Okay? So that we'll be able to differentiate Peptostreptococcus anaerobius from that of Peptonephilus asaccharoliticus. So, Peptostreptococcus anaerobius is said to be susceptible because there is a zone of inhibition. Pep, pep, Peptonephilus asaccharoliticus is considered to be as resistant because of the absence of the zone of inhibition. And lastly, among our special potency discs, we have the bile disc. So bile disc will somewhat assess the ability of the organisms to tolerate, okay, bile by growing at 20% bile because not all organisms will have the ability to grow in the presence of bile because bile is somewhat inhibitory. Okay, so it means that the organisms are bile tolerant. Okay, if they are resistant or if there is no zone of inhibition. Okay, so Gram-negative bacillus is most likely a member of Bacteroides fragilis. So they are said to be bile tolerant. But if there is a zone of inhibition, it means that they cannot grow in the presence of bile. So they are not bile tolerant. Okay, so other tests would include lecithinase and lipase reactions. Okay, so lecithinase is an enzyme, okay, that will cleave lecithin. So lecithin is found in egg yolk. So that's the reason why, in order for us to perform lecithinase reaction, you have to use an egg yolk agar. So what will happen is that the organisms will release an insoluble fat and it will form on a opaque zone around the colony. So usually that would be the characteristics of Clostridium perfringens. So this is a positive reaction for lecithinase reaction in egg yolk agar. Okay. Other one is lipase. So this one is another enzyme that would determine the ability of the organism or the enzyme to hydrolyze triglycerides and diglycerides to fatty acids and glycerol. Okay, so the triglycerides and diglycerides would be broken down into fatty acid and glycerol. So positive organisms are iridescent multicolored heme, such as the one with Fusobacterium necrophorum. Yan. So this is a positive reaction in lipase reactions. And then we also have the proteolytic reactions. So, what do you mean by proteolytic? Proteolytic is actually the ability of the enzyme to break down protein. And that would be possible because of proteolytic enzymes that we usually call them proteases. So, how do we know if there is a proteolytic reaction? So, there would be clear zone. So the presence of complete clear zone surrounding the colony. So if this is the colony, if these are the colonies, and then there are clear zone, so that is a positive reaction for proteolytic reaction. And it's quite difficult to observe them if the lighting condition is not good. So that's the reason why we have to hold the plate against a strong light source so that you'll be able to see a transparent plate here. It means that it is actually opaque. So it is reminiscent of the complete clearing zine with beta hemolytic organisms on SBA plate. So which means that the reaction is the same as beta hemolysis, except that we are not assessing hemolysis here, but we are assessing proteolysis here. Okay, so that is proteolytic reactions. Okay. Now, let's discuss the presumptive identification of gram-positive anaerobes. Okay, so these are the different examples of gram-positive anaerobes. So we have the Clostridium difficile. So they are large flat colonies, barnyard color, chartreuse fluorescence on blood agar. Cellular morphology, they are thin rod and rarely they have spores and they are negative in spot indole. Clostridium perfringens would have large irregular shape and double zone of beta hemolysis. So this double zone of beta hemolysis is also known as the target hemolysis. We will be discussing this one later on. So they have the boxcar large square rods, and we don't usually do spot indole for this particular organism. And then Clostridium septicum would have swarming. What do you mean by swarming? So if this is the blood agar plate, and then you place the culture here, the organisms will actually spread. The colonies will spread. Okay, so spreading of organisms in the plate is called swarming. So one particular bacteria that is very known for swarming is Proteus, but I will be introducing them to you during the finals. But anyway, Clostridium septicum are characterized by thin rods with subterminal spores, and they are negative in spot indole. Clostridium sordelli would have a very large, lobate, irregular, flat, and thin rods and subterminal spores. are the concellular morphology, and they're positive with spot indole. Clostridium tetani are smoothly swarming, but they are slow growing. So they have swollen terminal spores. So that is the reason why they are known as the tuck head bacillus. So they are known as the tuck head bacillus, and they're positive with spot indole. Peptostreptococcus would have small peak circular, and they are cocci that could either occur in pairs or in chains, and we don't usually perform spot indole tests for this particular organism. Cutibacterium acnes is characterized by small, opaque, enamel-white circular colonies, and we usually perform catalase tests for these organisms. So if you remember catalase test, rapid effervescence of gas is considered to be as a positive reaction. So this one is characterized by coriniform rods, and it is usually positive with spot indole. Okay, so these are the different presumptive identification for gram-positive anaerobes. So this is what you call the double zone of hemolysis, or otherwise known as the target hemolysis by Clostridium perfringens. Okay, so double zone because you have an alpha hemolysis and beta hemolysis that are adjacent to one another. So that's the reason why we call them as the target hemolysis or the double zone of hemolysis. Okay, so let us now discuss the presumptive identification of gram-negative anaerobes. For Bacteroides fragilis, um, we can either use, by the way, blood agar or KVLB agar. So Bacteroides would have large colonies, and the colonies on BBE agar, which stands for Bacteroides balesculin, would be large, convex black-gray, and the cellular morphology is regular, um, and spot indole is not being done. For Campylobacter urolithicus, they would have translucent and pitting in BAP or KBLB agar. KBLB agar, okay? Kanamycin, vancomycin, lake-shaped blood. So that's what we meant by KVLV. But they cannot grow in BPE, and it is characterized by tiny rods, cocobacilli, and it's negative in spot indole. Bilophila wordsworthia would have tiny translucent colonies in BAP and KVLB, and also translucent with black centers after 3 days in BBE agar. So the organisms are regular to filamentous, and it is negative with spot indole. For Fusobacterium nucleatum, it is characterized by ground glass or breadcrumb appearance, but there's no growth on BBE agar. So it is characterized by fusiform bacteria, thin with pointed ends, and is positive in spot indole. For Porphyromonas, um, it is small, translucent, or opaque with brick red fluorescence on blood agar and no growth on KDLP. They cannot grow also on BPE, but it appears like cocobacilli and it's positive with spot indole. For Prevotella intermedia, small, translucent, or opaque, brick red fluorescence on blood agar and KBLB. They cannot grow on BBE and they appear like tiny cocobacilli. Negative also for spot indole. For Veillonella, small, translucent, opaque, brick red fluorescence on blood agar, but there's no growth on KVLB. No growth on BBE. They are also appear like tiny diplococci and they are negative on spot indole. So again, um, these are very rare anaerobic bacteria, and some of them would really look similar as far as the colonial morphology is concerned, and that is the reason why we need further confirmatory testing in order for us to differentiate the organisms. But usually in the hospital, we usually correlate it with the specimen that was submitted to us, and of course the clinical history, as of course informed by the requesting physician. Okay, so these are the colonies of Bacteroides colonies on BBA agar, Bacteroides fragilis, and anaerobic KVLB agar, and BBE on our left. And then we also have the Bacteroides wordsworthia on BBE on our right. Okay. This is the gram stain of Fusobacterium nucleatum. So take note that you'd notice na medyo spindle-shaped branching yung morphology. Okay. So let's now discuss the definitive identification of anaerobic isolates. Yan. So for anaerobic identifications, there are several options. No? And most of these options are actually kind of expensive and not being done in the laboratory unless the machines and the kits are available. So noteworthy to mention is this one, the 16S rDNA sequencing, because this would involve PCR. Multitoph MS is actually practical because having this in the lab means that you can identify not just anaerobic bacteria, but other bacteria as well. And if it has MS, it means that it can also give you the result of susceptibility testing. So that's why it's a very good machine to have in the lab. But there are some presumptive identification. It says here, the one that we discussed a while ago, it has limited capability. So many of the clinically encountered anaerobes cannot be identified. For definitive identification, some lab would simply purchase commercially available kits or card system, such as the Iniden, RapidD, BBL, Vitec, and so on. Okay, so there are also commercially available biochemical micro identification system. Micro because you don't have to use several test tubes for this, such as the API-20A, the Minitech Sector. So these are examples of the minor uh, or mini identification biochemical based system. Okay, so we can use therefore biochemical based multi-system, preformed enzyme based system, cellular fatty acid by high resolution gas liquid chromatography. So matrix assisted laser absorption ionization or yung tinatawag natin Malditoff, and then 16S ribosomal gene sequencing. Ito na yung molecular na to, PCR na yung gagamitin. So these are for definitive identification technique. So this is what I told you a while ago, that we have the rapid preform enzyme system. So you will have to interpret each, and then there is a numerical what you call this, numerical equivalence of each of the reaction. And after you have determined the numerical equivalence of each of the reaction, you need to enter this number sequence in the computer software, and then the computer will tell you what organisms were you able to isolate. Okay, so this makes the life easier for medical technologies, no? Okay, so let's now discuss identification of Clostridium species. So for Clostridium species, we have several techniques, such as the double zone of hemolysis. I think I did mention this to you a while ago. The double zone of hemolysis is also known as the target hemolysis, usually useful for Clostridium perfringens. Do not forget this one. Um, so the inner zone is said to be a complete beta hemolysis, and then the outer zone is actually a partial beta hemolysis. Okay, so that's why it's called double zone of hemolysis. The CCFA is otherwise known as the cycloserine cephositine fructose agar. So this is the CCFA agar. And this one is useful for Clostridium difficile, selective and differential for Clostridium difficile. It will actually give you a yellow ground glass colonies, and it has a horse stable odor. So, amoy kulungan ng kabayo. So, that's how they describe it. And then, I think a while ago, we also have discussed the lecithinase and lipase reaction. Okay? So, usually, it says here the alpha toxin will be responsible for the lecithinase reaction for Clostridium perfringens. Okay? And then we also have the spot indole test. So I did mention to you this already, the spot indole. Blue-green is positive reaction for spot indole test. And then the rapid urease test, wherein the deep pink is considered to be as a positive reaction. So this one is manufactured by Kimberly Clark. It says CLO test. So usually it's a rapid test for Clostridium. That's why they call it the CLO test. And then we also have here the gelatin hydrolysis, the ability of the organisms to liquefy a gelatin agar. So that's the principle of the gelatin hydrolysis test. And of course, the gas liquid chromatography. So among these are routinely being done in the lab, in microbiology lab, and the gas liquid chromatography could be sent out to a specialized chemistry laboratory. Okay, so this one is the identification algorithm for some clinically encountered Clostridium species. Okay, the first thing that you have to determine is whether or not the blood agar plate has a double zone of hemolysis or target hemolysis. And if it does, then you're lucky because it means that you can now stop because you have identified Clostridium perfringens. So remember, only Clostridium perfringens among the clinically important Clostridium species will give you a positive reaction in the double zone of hemolysis. But if there's no double zone of hemolysis, then maybe you can do the lecithinase reaction on the egg yolk agar. Okay, let's now consider a negative reaction. If it's a negative reaction, then you have to do, this is the first test, second test, then you have to do the lipase reaction. If it's positive, then you're able to identify Clostridium sporogenes. If it's negative, then you have identified definitive negative, so you have to use a definitive methods specifically written in your book, particularly on table 22.9, which you can refer to me on later on. Okay, now let's talk about the condition if it's positive in lecithinase reaction. Okay, so again, you have to do the third test, which is the lipase reaction. Okay, if it's positive, then you are dealing now with Clostridium noyvi. Okay, then you do further test, which is the rapid urease test. If it's positive, then you're dealing with Clostridium sordelli. If it's negative, then you're dealing with Clostridium bifurmentans. Okay, but if you have a negative reaction in the lipase reaction, then you can do now another test. So this is the fourth test for this particular pathway. So if it's negative, spot indole test. I'm sorry, this pala is the fifth test, no? I'm sorry. Kapag positive pala sa lipase reaction, end of the algorithm. Because you now have the Clostridium noyvi, so I stand corrected. So you do first the fourth test, which is a spot indole test. If it's the positive, you do the fifth test, which is the rapid urease test. And if it's positive in rapid urease test, you're dealing with Clostridium sordelli. If it's negative, then you're dealing with Clostridium bifurmentans. Okay? Now, if it's negative in spot indole test, the fifth test that you have to do for this particular pathway would be the gelatin hydrolysis test. If it's positive, then you're dealing with Clostridium limosum. If it's negative, then you're dealing with Clostridium barati. Okay, so these are the algorithm. Okay, algorithm for clinically encountered Clostridium species. Okay, so this is the table a while ago that you need to study. So, yeah. So this is another example of double zone of hemolysis for Clostridium perfringens. So let us now discuss identification of anaerobic non-spore former gram-positive bacilli. So here we are dealing with actinomyces such as Actinomyces israeli, Actinomyces meyeri, Neslundi, Odolenticus viscosus, and some Bifidobacterium species, Egartella lenta, Eubacterium species, Cutibacterium acnes, and Propionibacterium propionicum. Noteworthy to mention are the colonies of Actinomyces israeli because it is the only one that are slow grower and has a molar tooth colony. Okay, molar tooth colonies. Okay, Bifidobacterium has forked ends. That's why it's called bifido. And Egertella is nitrate positive. Okay, so again, these are some of the organisms. So I think we have here the Actinomyces, Bifidobacterium that has a forked end, and then we have here Lactobacillus and Propionibacterium. Okay, so the forked end of Bifidobacterium is here. Okay, so this is the gram stain appearance of Actinomyces israeli and the molar tooth colonies of Actinomyces israeli. So the only one that has this kind of characteristics. Um, Bifidobacterium ranges from Coccobacilli to large branching rods. So ends may be pointed, bent, club-shaped, spatulated, bifurcated, forked. Okay, kaya siya tinawag na Bifidobacterium. Okay, the cells may appear singly or in chain and star-like arrangement and palisade. So when you say palisade, they are side-by-side arrangement. So the colonies, these are the colonies characterized by cream to white, smooth, glistening, soft colonies. Okay, so this is the Bifidobacterium. Okay, so when you say bifido, bifurcated fork. Okay, so that is the characteristics of Bifidobacterium. Okay, for Lactobacillus, the morphology varies greatly. So some species appear as pinpoint alpha hemolytic on blood agar plate, and others are medium in size with rough appearance and gray color, such as the one that you can find in this particular illustration. So they are catalase negative, and differentiation from Streptococcus species is difficult unless we perform gram stain. Obviously, Streptococcus are cocci, Lactobacillus are said to be a bacilli. Okay, so this is the gram appearance naman of Propionibacterium acnes. Okay, now let's talk about identification of anaerobic gram-negative bacilli. So under this, we have the Bacteroides fragilis, the Campylobacter urolithicus, the Parabacteroides, Prevotella, Porphyromonas, and Fusobacterium. So let's talk about the Bacteroides. So species are divided into two categories. Those that are considered to be as bile tolerant and those that are considered to be as the bile sensitive pigmented species. So those that are bile tolerant are the Bacteroides fragilis group because they can grow in the presence of 20% biles. The colonies on BBE and so ito yung colonies on BBE and robust growth in KBLB agar. So this is the BBE and then this is the KBLB agar. So bile-sensitive pigmented species cannot grow on BBE because they cannot tolerate the presence of bile. So these are the Porphyromonas and Prevotella. So this is the gram stain of Bacteroides fragilis. So they are gram-negative Coccobacilli and they are frequently pleomorphic. So when we say pleomorphic, it means that they can assume a variety of shape. So the organisms can assume a variety of shape, particularly in the broth culture. So Bacteroides fragilis culture, so this is how they appear on BBE. So they are gray and a minimum of 1 millimeter in diameter after 24 hours of incubation. Pale yellow of the culture plate turns brown to black in the area around the colony. So originally, they are pale yellow, but eventually, it will turn brown to black. So there is a dark precipitate or stippling in the medium around the heavy growth, and that actually suggests the presence of Bacteroides fragilis. Okay, so KBLB, this one is the KBLB, and this is the BBE agar. Okay, now for the bile-sensitive non-pigmented species, we have the Bacteroides urolithicus. So we isolate them from the blood following tooth extraction, and they are urease positive. Prebotella, on the other hand, may be isolated from vagina and oral cavities. So Prebotella denticola, okay, so they are escalin positive. Prebotella intermedia, okay, they are indole positive, lipase positive. So some biosensitive pigmented species can be categorized as pitting or non-pitting colonies. Okay. And then we also have the Bilophila wordsworthia. So the one that is noteworthy to mention here is the fish eye appearance of the colonies. And they can grow on BBE, okay, and KVLB. They are strongly catalase positive and strongly nitrate positive. Okay, Prevotella can produce protoporphyrin. So the reason why there is a brown-black color, okay, shift of macroscopic colonies for over 2 to 3 weeks. So they are either cocobacilli or bacilli gram-negative. So they can also produce a brick red fluorescence under long wave UV light, and they are resistant to kinamycin and vancomycin. So that's why they cannot grow in KVLD agar. So they are susceptible to colistin using the special potency test. So this is now the fluorescence that is associated with Prevotella. And then Porphyromonas will produce brick red fluorescence under long wave UV light. However, some species do not fluoresce. So they are typically susceptible to vancomycin. Therefore, they cannot grow in KVLB agar. But resistance to colistin is also seen with special potency tests. And most species are indole positive. So Campylobacter urolithicus contains bile-sensitive and bile-tolerant non-pigmented members. So they are microaerophilic. So what does it mean? So when you say microaerophilic, it means that they will only tolerate small amount of oxygen, but they are not obligate anaerobe. So colony appearance is helpful in identification. So they can grow in broth by adding formate or fumarate. Okay, so therefore, this particular broth is said to be an enrichment broth because you have to enrich first it with formate or fumarate. So members of Campylobacter urolithicus group are said to be nitrate positive. And we also have here Fusobacterium, generally considered as gram-negative, tapered rods, fusiform. So many appears pleomorphic. So again, when you say pleomorphic, so it means that they can assume a variety of shape. Okay? So they are classical fusiform shape. Okay, such as the one that you can see here. And they are indole positive, and they can produce butyric acid from peptonese glucose media or the PYG media. So two important species are Fusobacterium mortiferum, which appears pleomorphic globular forms. They have swelling and other bizarre shape. Fusobacterium mortiferum is indole negative, bile resistant, and esculin positive. And then Fusobacterium rusi is indole negative and bile sensitive. So here we have the Fusobacterium nucleatum on the left and Fusobacterium mortiferum on the right side. And of course, let us now discuss the identification of anaerobic cocci. So again, we have, this is the algorithm. So the first thing that you need to do is to check the color of the colonies, if it's black or not black. If it's not black, then the first test that you need to do is the SPS disc. If it's susceptible, then you're dealing with Peptostreptococcus anaerobius. If it's resistant, then you have to do the second test, which is the spot indole test. If it's positive, you have to do the third test, which is the nitrate reduction. So if it's positive, you are dealing with Peptonephilus, or formerly known as Peptostreptococcus indolicus. That's why it's positive for indole. And then if it's negative for nitrate reduction, then you're dealing with Peptonephilus, or formerly known as Peptostreptococcus asaccharolithicus. But if it's negative for spot indole, the third test that you need to do for this particular level would be the rapid urase test. If it's positive, then you're dealing with Anaerococcus tetradius, or formerly known as Peptostreptococcus tetradius. So tetradius, the reason why we call it tetradius, because cells occurs in clumps and in tetrads. Okay? But if it's negative, then you have to use biochemical or enzyme test or gas liquid chromatography to further identify the bacteria. But if you're dealing with black colonies, then maybe you're dealing with Peptococcus niger. They are weakly catalase positive and rarely isolated from clinical specimens. So most likely, you will have a non-black colonies. Okay. So this is the appearance of the anaerobic gram-positive cocci under the microscope. And these are some of the biochemical tests. Okay, so for the clinical encountered anaerobic cocci, noteworthy to mention, only Velonella parvula will have red fluorescence. Only Peptonephilus indolicus would be positive for nitrate reduction. Only Anaerococcus tetradius will be positive for urase. Only Peptonephilus asaccharolithicus and indolicus would be positive for indole. Only Veillonella parvula would be positive for catalase. And only Veillonella parvula would be negative in gram stain, because the rest are said to be gram positive. Okay, so the presumptive identification can be reported as Peptostreptococcus species or anaerobic gram-positive cocci. So we don't usually report the species if you're just dealing or doing the presumptive identification. The only way that you can report the species if you have performed the enzyme system, the 16S rRNA sequencing, and the MALDI-TOF MS. So Let's talk now, or let's discuss now the antimicrobial susceptibility of anaerobic bacteria. So we are actually being guided not just for anaerobic bacteria but for entire microbiology. We are being guided by CLSI, Clinical and Laboratory Standards Institute, and each year these particular guidelines are actually being revised. So perform when anaerobes are recovered from brain abscess, endocarditis, infection of a prosthetic device or vascular graft, joint infections, osteomyelitis, and bacteremia. So they are highly virulent or commonly resistant. Okay, the highly virulent or commonly resistant group. So we do not want to get infected with this. Bacteroides group, Prevotella, Clostridium, Fusobacterium, um, um, B. Wordsworthia, and Suterella Wordsworthensis. Okay? So this is an example of a spore-forming anaerobic bacteria. So for gram-negative anaerobes, the primary choice are the beta-lactam combination. Not just one, but a combination of ampicillin and sulbactam, amoxicillin and clavulanate, and piperacillin and tazobactam. I think we have already discussed um, the need of amoxicillin to have a to be combined with another substance such as yung augmentin. It's a combination of amoxicillin and clavulinic acid. So primary choice also would include clindamycin and the carbapenems such as ertapenem, imipenem, meropenem, and metronidazole. Supplemental choices would include um, cephalosporins, several generations such as ceftizoxime, ceftriaxone, cefoxitin, and moxifloxacin. For if you're dealing with gram-positive anaerobes, so primary choices are ampicillin and beta-lactam combination agent, such as the one that I did mention a while ago, clindamycin and carbapenem plus metronidazole. But supplemental choices would also have ceftizoxime. So that's the only additional here for gram-positive anaerobes. Okay? So there's a problem in susceptibility testing of anaerobic isolates because of the lack of reproducibility. It's quite difficult to have a precise results because of the difficulty of anaerobic condition. Failure of some anaerobes to grow appropriately because they're so sensitive with oxygen. Lack of comparability between methods, clinical significance of isolates. You cannot just do this on a benchtop, cost and complexity of testing. So CLSI standards have identified two methods. Either we do the agar dilution. So this is the agar dilution. This is a gold standard but is labor intensive, not practical for smaller clinical lab, primarily used in reference or larger laboratories or for research purposes. And then we can also perform the broth microdilution. This one is also important because we can determine the MIC or the minimum inhibitory concentration. So alternate method is the e-test. This is an example of the e-test. Here, we can also perform the MIC, or we can also determine the MIC. And some laboratories report out published susceptibilities that they do not do the testing in-house. So if, they are actually sending them out. So treatment for anaerobe associated diseases. So there are approaches to the management of anaerobic conditions. Surgical therapy is one, debridement, and then sometimes, unfortunately, they if it's too much gangrene, too much myonecrosis, they do amputation. Hyperbaric oxygen will create environment not conducive to anaerobic bacterial growth. Antimicrobial therapy, antitoxins to reduce virulence factors such as being injected with anti-tetanus toxin, and then fecal microbiota transplant. So this one is part of your online discussion assignment. So it involves the insertion via colonoscopy or endoscopy of donor feces into the into the patient. So if we have blood bank, we also have stool bank. Okay, so this ends the part 2 of anaerobic bacteria. So I hope everybody will study. So if you have any questions, please do not hesitate to reach out for me. Thank you, everyone, and God bless you all.